Hello everyone, hope you are all fine and doing great. Welcome to my class. Today I am going to start with another important topic from molecular basis of inheritance, that is packaging of DNA helix. Clear? The first question that comes to our mind, why packaging of DNA helix? What is the reason behind it? What is the basic concept? Now, from our previous discussion in the class, we know that DNA is a long polymer or chain of deoxyribonucleotides. The length of the DNA is far greater than the size of the area where the DNA is present. Therefore, there is a need of packaging of DNA so that the long sized DNA can be easily accommodated or incorporated in the small sized area of the cell. DNA is negatively charged because of presence of large number of phosphate groups. In the negatively charged DNA, packaging is done or packaging occurs by folding of the DNA, which is brought about by certain positively charged basic proteins. Now children, have a look. The note is there of this topic in brief. You can write it down and come to the further explanation of the process of packaging. Clear? The length of the DNA is calculated or measured by number of base pairs multiplied by the distance between two adjacent base pair or consecutive base pair. And you know that the distance between consecutive base pair from DNA double helix model is, can you tell me? Yes, that is 0.34 nanometer. Now this explanation is given here, you see, this is the nucleus of a diploid mammalian cell. Clear? The DNA is there, that is a human cell, that is 2.2 meter approximately. And this is distributed across all 46 chromosomes. Means if you isolate the DNA from this 46 chromosome, the value will come as 2.2 meter approximately. So the dimension of the nucleus is very small, that is 10 to the power minus 6 meter. Similarly, you see here in prokaryotic cell like E. coli, the length of the DNA is 1.36 millimeter. But the size of the cell where the DNA is present is very less, 1 micrometer. Clear? Here also you know, how can you find out the length of the DNA of the E. coli? By the same process I told you. And total number of base pair, you should remember. Can you tell me anyone from the class? Total number of base pair in the DNA content of the E. coli is 4.6 into 10 to the power 6 base pair. So from there, if you do the calculation, it will come as 1.36 millimeter length of the DNA. Now in both the cases, my dear children, both in human diploid cell as well as in E. coli, the prokaryotic cell, the length of the DNA is far greater than the area of the cell where the DNA is present. That's why there is need of, yes, packaging of the DNA. So as because the DNA is negatively charged, so the packaging by folding is done by certain positively charged basic proteins. Now children, separately we will be discussing in little bit detail about the packaging process of DNA in prokaryotes and eukaryotes. Now after conceptual explanation of packaging of DNA helix in general, let us study the DNA packaging in prokaryotes. Have a look. Now in prokaryotes, well-defined nucleus is absent. That is, there is absence of nuclear membrane or nuclear envelope. So even though the well-defined nucleus is absent in prokaryotes like E. coli, the DNA is not found scattered within the cell. Rather, the DNA is found in supercoiled state. The DNA is found in supercoiled state within the cell. And it is because the negatively charged DNA is held with certain positively charged proteins in an area called as nucleoid. The DNA in the nucleoid is organized in large loops held by proteins. These proteins in prokaryotes are non-histone basic proteins such as polyamines. The compact structure of DNA in prokaryotes is called as nucleoid. Now children, 2 or 3 questions only asked from this packaging process in prokaryotes in NEET and board examination in the form of correct or incorrect statement. Now first question is, that is, DNA in prokaryotes are found scattered within the cell. Is it correct or incorrect statement? It is incorrect statement because DNA is not found scattered within the cell of the prokaryote. Clear? Second question is, the DNA in the nucleoid is organized in large loops held by proteins. Is this statement is correct or incorrect, my dear children? It is a correct statement. Now DNA packaging in eukaryotes. It is much more complex in eukaryotes. My dear children, DNA packaging is much more complex than eukaryotes as compared to prokaryotes. This is a very important line of NCERT, that is much more complex. You have to remember this line for attempting question in NEET examination. The second point is, the process of packaging of DNA helix is brought about by a set of positively charged basic proteins called as histones. Remember the word histone. Okay, very important, my dear children. The second point. Third point is, histones are reaching to amino acids. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Have a look, my dear children. The spherical structure is histone octamer and the DNA is wrapped around the histone octamer. Wrapped means lapetna. Clear? Nucleosome is the unit of compaction or packaging in eukaryotes. Very, very important. Now recall, while discussing prokaryotes packaging of DNA, I have told you that the packaging unit of DNA in prokaryotes is, anybody from the class? Yes, that is nucleoid or genophore. A typical nucleosome contains 200 base pair of DNA. Very, very important. Very important, my dear children. How many base pair are there in a typical nucleosome? The answer will be 200 base pair. Okay, remember this thing. Next, nucleosomes form the repeating unit of a structure in nucleus called as chromatin. Chromatin is thread-like colored or stained bodies found within the nucleus. Clear? Very important, my dear children. Some of the lines, some of the word I have told you while discussing the packaging process in eukaryotes. Remember and by heart this for attempting NEET questions. Okay? Nucleosomes. The nucleosomes in chromatin are seen as beads on string structure when viewed or seen through electron microscope. इसका मतलब है, जब हम लोग इलेक्ट्रॉन माइक्रोस्कोप में न्यूक्लियोसोम्स को देखेंगे तो ये एक धागे पर मोती जैसा दिखता है. क्लियर? यहाँ बीड्स रिप्रेजेंट करता है न्यूक्लियोसोम को. बीड्स मीन स्मॉल बॉल लाइक और मोती जैसा स्ट्रक्चर है, वो रिप्रेजेंट करता है न्यूक्लियोसोम को. whereas योर स्ट्रिंग जो रिप्रेजेंट करता है, स्ट्रिंग मीन्स धागा, धागा जो रिप्रेजेंट करता है वो रिप्रेजेंट करता है क्रोमाटिन को. ओके चिल्ड्रन, द बीड्स ऑन स्ट्रिंग स्ट्रक्चर इज पैकेज्ड टू फॉर्म chromatin fibers. Clear? Next point. The chromatin fibers are further coiled and condensed at metaphase stage of cell division to form your chromosomes. Clear? And you know that metaphase stage is the best stage to study the structure of the chromosome, the morphology of the chromosome. Clear? Next, the packaged chromatin at higher level requires a set of additional proteins called as non-histone chromosomal proteins. Remember this point, very important. In a typical nucleus, chromatin shows two regions. Means the chromatin is differentiated into two parts. One is your euchromatin and other is heterochromatin. Differences between euchromatin and heterochromatin is given. Euchromatin is loosely packed, means ढीला रहता है euchromatin, whereas heterochromatin is densely or tightly packed. Euchromatin is light colored, lightly stained. Euchromatin is lightly stained, means light color का होता है. Heterochromatin is darkly stained, means dark color का होता है. समझ में आ रहा है? Stained means color. Stain का मतलब है color. Clear? Next. Euchromatin is transcriptionally active. Yes, anybody from the class? What do you mean by transcription, my dear children? DNA to RNA. So, euchromatin is transcriptionally active, while heterochromatin is transcriptionally inactive. These are the three differences given in NCERT. One extra additional point I am giving you. Histone proteins. Histone proteins are present in chromatin as well as in chromosome. Because chromosomes are the condensed form of your chromatin. So histone is present in both. But less histone, remember this thing, less histone in euchromatin and more histone in heterochromatin. That's why heterochromatin more histone, so dark लगता है देखने में. Whereas euchromatin less histone, so light color का होता है. Clear? Can you tell me who has given the name chromatin? You have studied in cell chapter. Chromatin are generally seen at interphase stage of cell division. Clear? Who has given the name chromatin? Yes, anybody from the class? Fleming. Yes, thank you. Fleming has given the name chromatin. Clear? One important question, children. Transcriptionally active, transcriptional inactive. Active is transcriptional active is euchromatin. Transcriptional inactive is heterochromatin. Clear? Now what about replication? DNA replication. DNA to DNA बनना को बोलते हैं replication. DNA replication और DNA dependent DNA synthesis. तो बोलो कि दोनों में से euchromatin heterochromatin में कहाँ replication होता है? In both euchromatin and heterochromatin, replication occurs at S phase of cell cycle. You have studied in cell division. DNA replication takes place at early S phase in euchromatin, whereas heterochromatin DNA replication takes place at late S phase. Very very important. Late S phase में होता है heterochromatin में. एक क्वेश्चन आया पहले NEET में. Clear? Now I will be discussing some important questions that are being asked in board and NEET examination. In addition to that, some of the important points and terms I will be highlighting. Now let us discuss some important questions from the chapter Packaging of DNA Helix. Have a look, my dear children. Question number 1, you see, how to find out the number of nucleosomes in the DNA of an organism. The simple point आपको याद रखना है, total number of base pair दिया होगा, divided by the number of base pair present in a single nucleosome. And I have already discussed a single nucleosome having got 200 base pair. Clear? It will divide by the total number of base pair present in the DNA of an organism divided by the base pair present in single nucleosome, then the result will come. This you remember. There are two questions given on the board. Have a look. How many characters are associated with heterochromatin? We know the characteristic of heterochromatin. Have a look. Number 1 is light color. It is wrong because heterochromatin is dark in color. Second point, you see, loosely packed. We know that is heterochromatin is tightly or densely packed. So it is wrong. Third number, that is major constituent of genome. Major main constituent of genome is right hai. Now you see transcriptional active. No, heterochromatin is transcriptional inactive. So it is wrong. Now replication, replicationally it is active. Both euchromatin and heterochromatin show replication. So it is right. Now non-inheritable. No, heterochromatin is inheritable from one generation to the next. Clear? Now replication at earliest stage. Heterochromatin does not replicate in earliest stage. euchromatin replicates at earliest stage of cell cycle, while heterochromatin replicates at late stage. So it is wrong. Out of all these statements given, 2 are correct. Which one? Major constituent of genome is correct and replicationally active होता है. Clear? ये दोनों statement इसमें से correct है, बाकी सब wrong है. इसलिए answer will be 2. Now come to the 4th question, my dear children, that is related to Watson and Crick model. That is double helix model is also called your 3-dimensional model. Remember this thing. Now correct steps in the organization of euchromatic chromosome. Clear or not? Eukaryote, the arrangement, the sequence of the packaging of DNA. You see, as per NCERT you have studied, that is DNA, then forms your nucleosome, nucleosome to chromatin, then chromatin to chromatin fiber. Chromatin fiber sometimes called as your solenoid. Clear? Next is your chromosome. Now come to the 6th number, types of histone. The types of histone, that is your 5 types, H1, H2A, H2B, H3 and H4. You see these are the different types of histone protein. Among these, the 4 types, H2A, H2B, H3 and H4, they form the histone octamer. These 4 histone proteins in double dose, they form the histone octamer. H1 is there. H1 is acts like a plugging protein. It is attached to the linker DNA. The nucleosomes are joined by DNA, that is called your, each nucleosome is joined by a DNA segment called your linker DNA, and H1 protein is attached to the linker DNA. Now come to the non-histone chromosomal protein. I have already told you that is the packaged chromatin at higher level requires such a set of additional set of proteins called your non-histone chromosomal proteins. The amino acid present in non-histone chromosomal proteins are, that is tyrosine and tryptophan. They help in packaging at higher level. Now histone proteins are the main thing that is given in NCERT. The amino acid present in histone proteins are, that is your lysine and arginine. These are all probable questions that may be asked in NEET and board examination, children. So remember all these questions and try to solve. Okay, thank you so much. Bye-bye.